So I am Sam Hall. I am doing my PhD research in Heather Stapleton's lab. And today I'm going to be talking about some of my doctoral research on PFAS in Pittsboro, North Carolina. So some of you may already be familiar with the story, but this made news in 2017, where Gen-X, a PFAS compound, was found to be contaminating the tap water in Wilmington, North Carolina. And so this made news because Gen-X is one of these compounds called a PFAS. So PFAS stands for per and polyfluoroalkyl substances, which basically means it has an alkyl chain of these carbon atoms put together, surrounded by these fluorine atoms, shown here in green. And what this carbon-fluorine bond does is it makes these chemicals really persistent. That's a very strong bond. So that leads to these compounds being called forever chemicals because they don't break down in the environment and they don't break down in the human body. About 5,000 different PFAS exist because they are really good at being stable and being used as water and grease-proof agents. Some of the ones that are more notable are these legacy compounds like PFOA or another one, PFAS. Those are commonly called the C8 chemicals. But now we're seeing more of these emerging PFAS, such as Gen-X, or some of these shorter chain compounds being used more. So PFAS are pretty much everywhere. Virtually everyone in the US has PFAS in their blood. We get exposed to PFAS through several different ways, such as by eating food. The food packaging has PFAS. Our drinking water can have PFAS. And the dust in our homes can have PFAS. And that's because PFAS are used in so many different products. So anything of your clothing or your carpeting or paper products that has a non-stick, waterproof, greaseproof coating likely has PFAS. In addition, firefighting foams can also have PFAS, and that's become an issue at certain military sites and airports that have extensively used firefighting foams. Those spills and the subsequent runoff has contaminated the water supplies and the environment near those areas. So in addition to being everywhere, these also have health effects. So some of the adverse health effects that we have found to be associated with PFAS exposure are things like higher cholesterol, deficits in the immune system like vaccine response, certain kinds of cancer, and reproductive and developmental toxicities. So we got interested in the community of Pittsboro, North Carolina, which is just outside the Durham area. It's in the same watershed as some other sites of PFAS contamination. So it's in the Cape Fear River Basin, upstream of Fayetteville and Wilmington. And it's near the Haw River. That's where the Pittsboro drinking waters river source is. And some of our research from our group has looked at water supplies in the triangle for PFAS. And when we were doing this, we noted that Pittsboro, North Carolina had much higher levels of PFAS in the tap water than the surrounding utilities. That's because Pittsboro is unique. It's the only one that draws its drinking water from the Haw River. And the Haw River, we found later, was contaminated with PFAS. Occasionally, these levels can get quite high. In our study a couple years back, we found levels as high as 760 nanograms per liter in the drinking water. As we take water from the Haw River, standard drinking water treatment doesn't remove PFAS from the water. So anything in the river is going to end up in the tap water, at least for PFAS. So because we noticed this in Pittsboro, we decided to do a much larger study. You can see here some of the study goals that we had. We had an aim of looking at what's in the river, an exposure study of looking at what's at the people, as well as like an animal study and some policy work. I'm going to be focusing on this exposure study, aim 2. So our goal was to recruit 50 Pittsboro residents and ask them to give us a sample of their blood and a sample of their water on the same day, so we could understand a little bit better how the PFAS in their drinking water is affecting the PFAS in their blood. And we actually did this at a couple different time points. We had seen before that levels of PFAS in the Hall River changed, so we suspected that levels in the drinking water could change. And we wanted to see if the levels of PFAS in their blood would subsequently change as a result of less or more PFAS exposure. And so before I go into what our results were, I want to really highlight how we communicated our results. So the first thing we did was we gave individualized reports to each participant, basically laying out what was in their own blood and water and how that compared to our study participants. We also set up a study website, so it was very clear what we were doing, why we were doing it, and what our results were and where they could go for future information. We also did a virtual town hall. We did this in October, so in the height of the pandemic, but we wanted a town hall sort of to be able to explain to the community what we were finding and to give them a space to ask us questions about the research that we were doing. And so we did this via Zoom and Facebook Live. Shout out to Sam Cohen and the Superfund team for helping us pull that off in partnership with the Hall River Assembly. It was really successful and really great way to explain what we were doing to this community. All right, what we found basically was, to nobody's surprise, that PFAS was in the drinking water samples of these participants. What's unique about Pittsboro specifically is that some of these shorter chain, newer PFAS are really present in the water. So in purple and blue here, these are the C6, C5, and C7 compounds. Typically the ones that we've heard of historically are the C8 compounds, PFOA and PFAS. Those are the ones that are in the movies, like Dark Waters. We're finding in Pittsboro more of these shorter chain compounds. In addition, we found a wide variety in the levels that we saw in the water. So while some people had high levels up to about like 450 nanograms, other people didn't have any PFAS in their water. And those are the people who were mostly using filters, like reverse osmosis filters for their water. Moving on to the blood results. This is a simplified version of the blood results that we have. This is just the median levels. But for PFAS and PFOA, even though it's not really high in the drinking water now, like presently in 2020 and 2019, we found it at higher levels in Pittsboro residents than in the general US population. So here in the gray, you can see what is typically found in the US population, and the red and the blue are the two different time points from Pittsboro. We didn't really see a change from the different time points for those chemicals, and that's not surprising because PFOA and PFAS have half-lives of several years. So we don't really expect those to change over the course. And also, if we go and look at more of these compounds in comparison to other communities, we can see what we see in Pittsboro is similar to what is found in Wilmington, North Carolina. So the median levels in Wilmington are pretty much equal to the median levels in Pittsboro. So some future directions. We have lots of future directions for this community and for this research on PFAS in general. But some things that are coming out pretty soon are the results from this rabbit study. We obviously can't do studies in humans to really tie down this association between PFAS exposure and health effects. So instead we do that in animals. And so that's what we've done in this rabbit study is we've exposed pregnant rabbits to drinking water concentrations that mirror what we see in Pittsboro. So an environmental mixture at the exact same levels to see what health effects do we see, and mechanistically, where did the PFAS end up in the mom and in the baby to give us an idea of what's going on in the actual animals. And then we are doing some further work on the Pittsboro blood samples. We're going to look at some clinical endpoints like cholesterol and some liver and kidney enzymes. And we're going to see if PFAS levels are associated with any outcomes in that serum. And so I'd like to for sure acknowledge our study participants. We couldn't have done the study without them, and all of our other partners that have made this possible. And with that, I will take any questions.